Good afternoon to all the EAPCET aspirants. So here we are starting out the major chapter, that is molecular basis of inheritance, where we are going to deal with all the important concepts of this chapter which are important for our exam. First we are going to begin with DNA structure, how a polynucleotide chain is formed and what are the bonds that are involved in single stranded DNA and double stranded DNA. Followed by we are going to learn about the salient features of DNA, Chargaff's rule, and then packaging of the DNA. Packaging of DNA. And then comes the discovery questions. Like discoveries, and the scientists will be asked mostly in the match the following type of questions, where we have to learn clearly about transformation principle by Frederick Griffith, followed by we have to also learn the Hershey and Chase experiment and biochemical proof that DNA is the genetic material. These three experiments we have to go through, and so it is followed by discoveries, and then we have to go through RNA world. Okay, we have to go through RNA world, followed by we'll be learning out after RNA world about replication, where we have to learn the experimental proof about semi-conservative method of DNA replication, that about Michelson and Stahl's experiment, followed by transcription and translation. Before translation, we have to also go through genetic code, and after translation, gene expression is most important topic, where we have to learn this with an example of lac operon. Fine, right? So these are the major concepts of molecular basis of inheritance. And then if you see occurrence of DNA, how DNA is present both in prokaryotes and eukaryotes. In prokaryotes, mostly the DNA is in the form of double stranded circular DNA or single stranded circular DNA, but DNA mostly exists in the form of circular naked DNA. Okay, and this DNA is not encoded by any nuclear envelope. At the same time, it is not scattered all across the cytoplasm. It is at one particular position, where you will call it as what? Nucleoid. And along with the genomic DNA, you also have non-genomic DNA, which is also addressed as extra chromosomal DNA, also noted as plasmid DNA. Okay, this is also known as plasmid DNA. And in eukaryotes, if you see, DNA structure is linear. It is double stranded linear DNA. It is found inside the nucleus, double stranded linear DNA is present inside the eukaryotes. So this is about prokaryotes and eukaryotes, how DNA is present. And we know DNA is a polynucleotide chain. Yes or no, ma? DNA is a polynucleotide chain. DNA is a polynucleotide chain. So this polynucleotide chain, each nucleotide, if you consider, okay, each nucleotide consists of a sugar molecule. Okay, nucleotide, whether it is over DNA or RNA, based on that, if it is over DNA, the sugar that is present is deoxyribose sugar, which is a pentose sugar. Okay, it is a pentose sugar. Okay, pentose sugar, which is deoxyribose sugar, which is at second carbon there is no OH group. At C2 carbon there is no OH group. Okay, which is a pentose sugar. To this pentose sugar, which is having 5 carbons, C1, C2, C3, C4 and C5. It has 5 carbons. At second carbon there is no oxygen group, that is why this is known as deoxyribose sugar. To the first carbon, nitrogen base is attached. This nitrogenous base is of 4 types if it is in the DNA. You have both purines and pyrimidines. Purines and pyrimidines. Purines can be adenine and guanine. Pyrimidines are cytosine and thymine. Right, ma? Wherein purines are heterocyclic ring. Pyrimidines homocyclic ring. Okay, purines are heterocyclic ring. Pyrimidines are homocyclic. Single ring is present. So this heterocyclic structures, it is having 9 nitrogen groups. So if the link is in between C1N9 of purine, which forms a glycosidic bond from sugar, carbon 1 of the sugar is binded to N9 of the nitrogenous base, okay, which is N-glycosidic bond. This is N-glycosidic bond. If it is pyrimidine, C1 of pyrimidine is linked with N1 of C1 of the sugar is binded with N1 of pyrimidine. Okay, C1 of the sugar is binded with N1 of pyrimidine. Okay, C1N1, which is of what connection again? N-glycosidic bond. C1 carbon of the sugar is binded to N1 of the nitrogenous base. Remember that. So sugar along with nitrogenous base is known as nucleoside. Okay, nucleoside includes sugar along with nitrogenous base. Okay, it includes sugar along with nitrogenous base. At 5th carbon of the sugar, phosphate group is binded. Okay, to the 5th carbon of the sugar, phosphate group is attached. This is by phosphoester bond. Phosphoester bond. So remember students, for a single nucleoside, what bond is required? N-glycosidic bond, which is C1N9 of purine, C1N1 of pyrimidine. Right, ma? And to the 5th carbon of this nucleoside or the sugar molecule, phosphate group is attached by phosphoester bond, and then it forms out what? Nucleotide. This is how a complete nucleotide is formed. In a nucleotide, 2 bonds are involved. One is N-glycosidic bond, second is phosphoester bond. For nucleoside, only single bond is involved, that is N-glycosidic bond. Clear till here? And here you see length of DNA in different organisms. So different organisms if you consider, first bacteriophage lambda into 174. This lambda bacteriophage has how many nucleotides? 5386 nucleotides are present in a single stranded DNA. In a single stranded DNA. Fine, right? How many are present? 5386 nucleotides are present. Next, bacteriophage lambda. Bacteriophage lambda, it has 48,502 base pairs are present. Okay, means see, if at all only nucleotides are present, it denotes that it is a single stranded DNA. Okay, if it has base pairs, then it says that it is a double stranded DNA. Fine, right? And then Escherichia coli will be having 4.6 into 10 to the power of 6 base pairs. Haploid content of human DNA will have 3.3 into 10 to the power of 9 base pairs. This length of the DNA is always calculated with number of nucleotides. Remember that. Okay, so here you can see both single stranded DNA and double stranded DNA. So you can see here 2 terminus. At 5' end will be free at the one end and 3' end will be free at the other end. 5' end is having phosphate group, 3' end is having hydroxyl group. Phosphate group at one end and hydroxyl group over the other end. And what are the bonds that are involved in single stranded DNA? If you see, in a single stranded DNA, you can see N-glycosidic bond is involved in between nitrogenous base and sugar. Phosphoester bond is involved in between nucleoside and phosphate group, and to connect 2 nucleotides here, okay, two nucleotides are connected at 3' end OH group and 5' phosphate group of the next nucleotide. 3' OH group of one nucleotide is linked up with 5' phosphate group of the succeeding nucleotide. This is by what bond? Phosphodiester bond. So in a single stranded DNA, three bonds are involved. Single stranded polynucleotide chain will have 3 bonds. Okay, one is N-glycosidic bond, second is phosphoester bond, and third is phosphodiester bond. Fine, right? Next, in a double stranded DNA, along with these three bonds, you will see in a double stranded DNA that backbone of the DNA will be having sugar phosphate groups. Internally the DNA will be having nitrogen bases. These nitrogen bases are connected to each other by hydrogen bonds. Okay, nitrogen bases towards inside, they are connected by what? Hydrogen bonds. It is two hydrogen bonds in between adenine and thymine, and 3 hydrogen bonds in between guanine and cytosine. Clear, right? So these are the bonds that are involved in single stranded DNA and double stranded DNA. Now we will come across the different nitrogenous bases. So as we have discussed, in RNA you will be having uracil and cytosine. In DNA you will be having cytosine and thymine. In place of thymine you will have uracil in RNA. Thymine in DNA in the place of uracil will give additional stability to DNA. These are pyrimidines, and these heterocyclic ring structures are what? Purines. It is important for you people to know the IUPAC names of the nitrogenous bases as well. So here if you see the common name adenine, the IUPAC name for this is what? 6-aminopurine. Okay, IUPAC name for this is 6-aminopurine. For adenine it is 6-aminopurine. Guanine it is 2-amino-6-oxypurine. Guanine 2-amino-6-oxypurine. Cytosine 4-amino-2-oxypyrimidine. Fine, right? Thymine 5-methyl-2,4-dioxypyrimidine. Uracil 2,4-dioxypyrimidine. Clear, right? So here you see adenine 6-aminopurine, guanine 2-amino-6-oxypurine, cytosine 4-amino-2-oxypyrimidine, thymine 5-methyl-2,4-dioxypyrimidine, uracil it is 2,4-dioxypyrimidine. Clear, right? So these IUPAC names you have to remember for sure. Next, these are the differences between purines and pyrimidines we have already discussed. If you want, you can take a screenshot of this and remember. Next. Yeah, these are the different names of nucleosides and nucleosides both in DNA and RNA based on the nitrogen base it has. Adenine plus sugar forms what? Adenosine. Guanine plus sugar, guanosine. What is this sugar? Ribose sugar. How can you identify that it is a ribose sugar? By having OH group at the second carbon. If it is having OH group at the second carbon, it is what? Yeah, just a second. Yeah, OH group at the second carbon, OH group having OH group at the second carbon, this is what? Ribose sugar. Ribose sugar. Fine, right? Ribose sugar will have OH group at the second carbon. Next, cytidine. Cytosine and ribose sugar is cytidine. Now it comes to thymine. Thymine along with deoxyribose sugar. At second carbon there is no OH group. So this is deoxythymidine. Adenosine along with deoxyribose sugar, deoxyadenosine. Guanine along with deoxyribose sugar, deoxyguanosine. Cytosine along with deoxyribose sugar, deoxycytidine. The names are very important. We got quite a times in our exam. You should answer it clearly. Okay. And now here we have to see for nucleotide. In a nucleotide, what happens? Phosphate group will attach to the fifth carbon of the sugar. Okay. In that case, this is for RNAs. First we'll see. This is adenylic acid, as phosphoric acid will be attaching to this, and phosphate group over the nucleotide will give it what? Negative charge. That is why DNA is what? Negatively charged. Guanosine plus phosphate group is guanylic acid. Okay. Adenosine plus phosphate group, adenylic acid. Cytidine plus phosphate group, cytidylic acid. Deoxythymidine, right? Deoxythymidine plus phosphate group, deoxythymidyllic acid, deoxycytidylic acid, deoxyguanylic acid, deoxyadenylic acid along with phosphate group. Clear, right? These are the names of different nitrogen bases being attached to the sugar, and for these are the names with different nitrogen bases for nucleotides as well. Fine, right? I hope this is clear. And then about double helical model of DNA, which is proposed by Watson and Crick. First, how nucleus is being discovered, how DNA is being discovered. It is discovered in 1869 by the scientist Frederick Mescher. He discovered the DNA as some acidic substance and named it as what? Nuclein. Later, Rosalind Franklin and Maurice Wilkins, they have gave out the X-ray diffraction pattern of DNA. In 1953, it is the scientists Watson and Crick, they have proposed the double helical model of DNA. This was based on the findings of what? Erwin Chargaff. Erwin Chargaff have given this. This is also known as what? Chargaff's rule, and this Chargaff's rules were based on what? X-ray diffraction studies. Erwin Chargaff has pointed out that in a particular DNA molecule, the total number of purines will be equal to total number of pyrimidines. Total number of purines, that is adenine plus guanine, is equal to total number of pyrimidines, that is thymine plus cytosine in a DNA. And do remember, they have in double helical model of DNA, it is also given that adenine plus guanine by thymine plus cytosine are always 1. They are constant in any species. This ratio is constant in any species. Fine, right? The number of adenine thymine bonds will differ from the number of guanine cytosine bonds, but the base ratio, that is adenine plus thymine by guanine plus cytosine, this is addressed as base ratio. So purines by pyrimidine is always constant in all the species, but the base ratio, that is adenine plus thymine by guanine plus cytosine, base ratio differs in each individual. It will be less than 1 in prokaryotes, approximately 0.9 in E. coli, and more than 1 in eukaryotes. Let's say for human cell only, it will be around 1.3% as such. These are the ratios. Fine, right? Remember that carefully. And then about silent features, we have silent features of DNA. We have different forms of DNA, A form, B form, Z form as such. but the standard form of the DNA is B form DNA. B form DNA is the standard form of DNA. It is made up of two polynucleotide chains where sugar for phosphate moieties will be backbone of the DNA. They will be facing outwards. Internally, nitrogen bases of two strands will be connected by hydrogen bonds. Okay. They are coiled in what fashion? Right-handed fashion. Okay. The pitch of the helix will be 3.4 nanometer. What is pitch? One pitch is equal to one turn of DNA. Pitch is one turn. One pitch is one turn. One complete turn of DNA is one pitch. Okay, this is one pitch or one turn. How much length it is? 3.4 nanometers. One pitch or one turn will have 3.4 nanometers helix. Okay, and there are around 10 base pairs for one pitch or one turn of the DNA will be having 10 base pairs in each turn. Consequently, the distance between two base pairs. Okay, so in a complete one pitch, in one complete turn, we have 10 base pairs, and the distance between two base pairs is, how much? 0.34 nanometers or 3.4 angstroms is the distance between two base pairs, and in a one complete pitch, the distance is what? 3.4 nanometers, which is 34 angstroms. Remember that. Okay, from nanometers to angstroms, that will be easy for your calculations. Remember that. The plane of one base pair stacks over the other in a double helix. This addition to hydrogen bonds confers the stability of helical structure. This confers the stability of the helical structure. Now, two chains will always run anti-parallel to each other. One strand runs from 5' to 3' and the other from 3' to 5', and the strands are complementary to each other. Okay, they are complementary to each other. The bases in two strands are paired through hydrogen bonds, forming what? Base pairs, and the purine always comes opposite to the pyrimidine. This generates approximately uniform distance. This generates what? Uniform distance between two strands of the helix. Now we will look into the packaging of DNA. DNA is being packed. So here you understand this carefully. DNA is around 2 meters long, and that long DNA has to fit into very short nucleus, which is around 10 to the power of -6 nanometers. Okay, so when this long DNA has to fit in that short space of nucleus, DNA has to be packed and coiled tightly. That will be done by histone proteins, which are positively charged. So this doesn't happen in a single step. First, double helical DNA is formed when DNA is attached with histone proteins. This is formed first by supercoiled DNA. First it turns into supercoiled DNA, then chromatin fiber, then DNA get attached to histone molecules, and later what? Double helical DNA is formed. How is this formed? You see, first DNA which is negatively charged is wrapped around positively charged histone proteins. Wrapped around histone proteins. These histone proteins are positively charged. Fine, right? And histone proteins are of 4 types: H2A, H2B, H3 and H4. These are the 4 types of histone proteins. They are present in 2 sets. Here in this diagram it will be clear for you. Yeah, histone octamer, it has H2A, H2B, H3, H4. these are present in 2 sets. Okay, 2 sets of this. This histone octamer, it will be having, this histone octamer is being wrapped by what? DNA, which is negatively charged. As such, the negatively charged DNA is compressed or condensed in association with positively charged histone octamer. In one histone octamer, how much length of DNA is wrapped, if that is the question? The answer should be 200 base pairs of DNA. 200 base pairs of DNA will wrap around the positively charged histone octamer. And from one octamer to the other, here in this diagram you can see, yeah, when DNA at the time of wrapping around the histone octamer, from one octamer to the other, the DNA is being linked, and this DNA is known as linker DNA, which has around 80 base pairs. And here it is how? 200 base pairs. Linker DNA is associated with H1 histone proteins. Like that, the DNA packaging will take place. Okay. And when it is packed, after packing, you can see the DNA which is condensedly, tightly packed, that regions of DNA is known as heterochromatin. This heterochromatin regions of the DNA are transcriptionally inactive. They are transcriptionally inactive. And euchromatin, these regions which are loosely packed, they are transcriptionally active. They are transcriptionally active, and these are inactive. Clear, right? And then search for genetic material. Here we have transforming principle, which is given by Frederick Griffith. Okay, in this experiment he has chosen the model organism, that is Streptococcus pneumonia. Okay, Streptococcus pneumonia. 2 strains of the bacteria, that is Streptococcus pneumonia, one is S strain and the other is R strain. S strain is known as virulent strain because the bacteria is coated by a shiny wall layer outside it. The bacteria is coated by a shiny wall layer outside it. Fine, right? This is virulent, where after entering into the host cell, the bacteria will still survive because it has a shiny wall layer over it which protects the bacteria. Whereas R strain will just have a wavy coat over the bacteria. There is no protective shield where it gets protected. So here if you see, first in the S strain, where you can see the virulent strain, living R strain, non-virulent strain. Living S strain, where the mouse dies. Okay, when living S strain is injected into the mouse dies because the strain is alive inside the mouse, which is active because it has a protective coat over it. Whereas the living R strain, which is injected into the mouse is alive and the bacteria deads inside. Fine, right? And when this virulent S strain is heat killed, when it is heated to high temperatures, the cell wall layer will be degraded completely and the mouse is alive. When the heated S strain is mixed along with the live R strain, along with living R strain, when this heat killed S strain is mixed, when this heat killed S strain is mixed along with the R strain and injected into the mouse dies again. Okay, the mouse dies again. Fine, right? Understood. So with the last experiment, when heat killed S strain is added along with R strain, which is making R strain virulent here, with this Griffith has confirmed that there is something that is being transferred from R strain to S strain, which is making the R strain virulent and mouse die. Fine, right? So, but what is that actually transforming is not being identified. That is being identified by further experiments, which is given by Avery, MacLeod and MC Cartier. they have given out the biochemical characterization of the transforming principle, and they have clearly explained that what is the material that is being transferred. So from the heat killed S strain and R strain, it is being taken in 3 different test tubes, okay, and in 3 different test tubes they have been taken. In one of the test tube, proteinase is added. Proteinase will digest proteins. Fine, right? Proteinase will digest proteins. So there is no degradation of the S strains here. S strains are appearing. And to the next test tube, RNase is being added. RNase will degrade RNA. Okay. And here still the S cells appear in the strain even after RNase is being added, because it doesn't affect the genetic material. When the DNase in the last test tube, when DNase is added, DNase will degrade the DNA structure. Okay, it will degrade the DNA structure. Thereby, when DNA is being degraded, after this if you see, the mixture doesn't have S cells anymore. The transferred cells are not present anymore. So, when DNase is degrading the DNA and later you cannot see the S cells, this has proved that DNA is the genetic material that is being transferred from heat killed S strain to R strain and made R strain virulent. Clear, right? This is about the biochemical characterization. And later it is Hershey and Chase who has given out the unequivocal proof that DNA is the genetic material. Okay, unequivocal proof that DNA is the genetic material. So here if you see clearly, first is what? Bacteriophage. Bacteriophage will have 2 structures or 2 components in it. One is protein component, and the second is, one is what? Protein component, and the second is DNA structure. Okay, DNA is present inside. DNA is present inside the protein capsid, and the DNA is double stranded circular DNA over which the protein coat is present, which is capsid. Fine, right? So here over one set of bacteriophage, the protein coat is labeled with a radioactive isotope, that is sulfur. Because the protein coat will have amino acids, sulfur containing amino acids, it has sulfur with a labeling S35, radioactive label. Next is radioactive P32, okay, isotope is being labeled for DNA for the other set of bacteriophages. To one set, the protein were labeled with S35, radioactive isotope. To the other set, the DNA is labeled with the radioactive isotope, that is P32. Clear, right? So when this is injected to the bacteria, first it is causing infection. When it is causing infection to the bacteria, and later when it is blended, first stage is infection, next is blending, post which centrifugation. After centrifugation, what happens is, in the test tube post infection and after centrifugation, the filtrate particles will be above in the supernatant. In the supernatant, the filtrate particles will be above, and below you can see the dense molecules which are present. Below you can see the dense molecules which is retentate. This is retentate. Below which the dense molecules are present is the retentate. So here if you see, after centrifugation, in the supernatant you can see only the particles, protein particles which are being dispersed, and in the retentate you can see the dense material, that is DNA structure. Okay, here you can see the dense material, that is DNA structure. So radioactive isotopes were detected in the supernatant post centrifugation, and here in the supernatant there is no radioactive isotopes that are present, where the DNA is labeled with P32 isotope. They are being entered into what? Bacterial genome. Okay. So with this they have confirmed, they have given the proof that DNA is being entering into the bacterial genome. Fine, right? And proteins will be present at the supernatant which are dispersed out. So DNA is being transferred to the next generation or to the organism. That is being proved by Hershey and Chase experiment. Next. Here we will learn about central dogma of molecular biology. Yeah, central dogma of molecular biology, which is given by Francis Crick, which says that DNA first undergoes replication, which is copied to another strand and then transcribes its information to RNA, post which the information is translated to proteins. The ultimate goal of DNA is to synthesize proteins. So first, for DNA replication, the major enzyme that is required is DNA dependent DNA polymerase. First we will see in the prokaryotes. Where does this happen? The entire process of replication, transcription and translation will happen in the cytoplasm of the prokaryotes itself. First, if you see the replication is done by DNA dependent DNA polymerase. DNA dependent DNA polymerase. Fine, right? And then when DNA is transcribed to RNA, fine, right? Here the enzyme that is required is DNA dependent RNA polymerase. Major enzymes I am writing, we will have another set of enzyme machinery, around 7 to 8 enzymes are required for DNA replication, but the major enzyme for the synthesis I am describing here. For replication, so it is DNA dependent DNA polymerase, for transcription it is DNA dependent RNA polymerase. Okay, and later is what? Translation. This information over the RNA is translated to what? Proteins. Fine, right? And then if you observe in eukaryotes, what happens, ma? First, DNA replication, transcription and post-transcriptional events which includes capping, 5' capping, second 3' splicing, 5' capping, 3' polyadenylation, and splicing. Till post-transcriptional events, all the events will occur inside the nucleus. Fine, right? After post-transcriptional events, protein synthesis or translation into proteins takes place inside the cytoplasm. This occurs inside the cytoplasm. So first DNA replication followed by transcription, post-transcriptional events takes place inside the nucleus and translation occurs in the cytoplasm of the eukaryotic cell. In few viruses where RNA is the genetic material, what happens is RNA will be reverse transcribed to DNA. That is reverse transcription, or it is also known as teminism. For that, what enzyme is required? RNA dependent DNA polymerase is required for reverse transcription. Remember that. RNA dependent DNA polymerase is required for reverse transcription. Clear, right? Replication, transcription, translation. From RNA to DNA, that is reverse transcription. What enzyme is required? RNA dependent DNA polymerase. Clear till here? Any doubts anywhere, ma? Yeah. So about replication, transcription, translation we will discuss in the next class, followed by the objective practice of EAPCET. Clear, right? So that's all for today's session. Revise everything thoroughly and get ready for the next class.